So, thank you, Ian. I hope my voice will carry all the way. I will do my best. I unfortunately got some cold coming. It's a great pleasure to be here, and I'm very happy for the organization and Richard, Pia, everybody who has been helping out. It's been super interesting for me also to follow. I will give you a bit of a tutorial on what we are doing and what is proteomics. And I think we can maybe help in this field by looking at large-scale protein analysis. I'm situated in Uppsala, Sweden, and at Uppsala University. And my share is in analytical chemistry and neurochemistry, and then I'm also adjunct professor in pathology in US. So let's start with analytical chemistry. What is analytical chemistry? Well, so Lord Kelvin actually defined that if we can measure something, it's very important. And if we can't measure it, we will not be able to improve it. And I think that's sort of our motto in analytical chemistry, that we will try to be as good as possible to measure, to be able to understand and explain things. If you look into the British science, William Harvey already in 1628 in his anatomical book actually described this phenomena a bit. And he did it in a very nice panel that I think if you have a chance to look at this anatomical book, you should go and see it. He describes the liquids or the body fluids and the color of the body fluids, and I hope most of us have fairly normal colors here, but you can see urine can come in many different colors, blood also, and gall and so on. So depending on the color, you could of course tell something about that something is wrong with the body. Nowadays we have these kind of assays, simple assays. You pee on a stick and you get information on proteins in urine, for instance, or glucose or so on. However, in order to do real analytical chemistry, you have to be a bit more sophisticated. And this was actually discovered in Uppsala for the first time in the 1700 by Torbjörn Olof Bergman. And he's my predecessor on my share in Uppsala. And he turned out to be the first analytical chemist in the world, actually. If you read a bit about him, he has an interesting story. He grew up in south of Sweden. His parents and himself agreed that he was not very good in school. He was a rather messy kid, actually. So they sent him to Uppsala to read theology, and he was super miserable with that. But another professor in Uppsala was actually Carl von Linnaeus, who made the Systema Biologica. And he recognized this Torbjörn. He was an interesting guy, and he sneaked into the lectures of Linnaeus and actually discovered some new insects, for instance. But then when the share was open in chemistry, he wasn't a chemist from the beginning. The king and Carl von Linnaeus helped him to get the share. And on that share, he actually made fantastic discoveries in analytical chemistry and chemistry. So he discovered and made carbonized water for the first time in the world. So you can say he sort of started Coca-Cola company then, which is a fortune. So in my laboratory, we do a lot of clinical research, and we mostly focus on the central nervous system and the brain. But we have nowadays applications in many different regions of the body, so we do everything from respiratory disorders to immune-related diseases, cardiovascular diseases, reproductive disorders, and gastrointestinal diseases. So a lot of the applications we have is actually based on our technology and the possibility to measure and to decipher the chemical pattern in a sample. We use a technology called mass spectrometry, and I will tell you a bit what that is. But first we will see what molecules are present in a biological sample. Well, all of you know about DNA and nucleic acids, but you know also that the result of those molecules are the proteins, and we have the carbohydrates, we have the low molecular weight compounds like metabolites and lipids. And all of these make our organism function, and if there are disturbances in certain levels of these molecules, we get into trouble. So if we are doing proteomics then, what is that? Well, as I said in the beginning, it's a large-scale analysis of all proteins. And it's defined as identifying the molecules of life. So what proteins are present where, when, how, and why. We would like to know also together with whom. So what other proteins are present at the same time. So you can say that's a sort of a holistic biology or a system biology approach to a compound or a protein in this case. Taking you back to school a bit, you remember in school that you learned about the cellular systems, and cells are very complex, organelle-rich compartments. And you know that we have energy organelles like mitochondria, we have nuclei, of course, with our DNA and nucleic acids. We have other organelles here that are very important. And the human genome contains about 26,000 genes. And we replicate the genome by transcription and translation, so we get the proteins out. How come we can have more than 1 million proteins if we only have 26,000 genes? Well, that's actually due to this part of the cell. It's called the Golgi apparatus. So it's a membranous compartment in the cell. So when the proteins are produced on the ribosomal complexes, they will be transported through the Golgi apparatus, and then we mature to proteins of different forms, so proteoforms, as it's called. One thing that will happen is that they will be modified, so we will have post-translational modifications. And we have nowadays a list of about 450 different modifications that are occurring in humans. And I've listed a few of them here. These modifications add on to the sequence of the proteins. And you can see, for instance, this region here of the protein can have a multitude of different modifications at the same site. So that is actually what makes us so complex, so the protein gets different biological function. You can measure this with traditional methods like two-dimensional gel electrophoresis, where you first separate your proteins in isoelectric focusing and then with size, and you create these two-dimensional pictures of the protein content. However, we are unfortunately not being able to use this for diagnosis, so it's not as easy as this. If we have a sample from a certain patient group, we will get a clear-cut answer. Unfortunately, it would be beautiful. So we have to use somewhat more sophisticated technologies today. And what we are doing is shotgun proteomics. So we take the whole sample, we cut it into small pieces by using an enzyme called trypsin, the same enzyme you have in your gut. We cut the proteins into small peptides, and then we separate them in a multidimensional way. And all these peptides will then be detected by sophisticated measurement instrumentation called mass spectrometry. And these mass spectrometers will generate pictures of the protein content, and these pictures can be compared among patients and controlled, for instance. We will know what protein is present, how much of it is there, if it's modified, as I said, the post-translation modifications, if they occur or not. Mass spectrometers look like, rather expensive household equipment, I would say. They come in different flavors, and they can do different things. The most sophisticated ones are the high-resolution mass spectrometers that we use for complex samples. We can have it analyzing intact proteins like this. So we have an intact protein injected into the instrument, and then we can pull the protein apart to look at all the details. And some instruments can be used, for instance, for dry blood spot screening. So you leave a small sample droplet of your blood from your finger, leave it to dry on a sample holder, and then you do the analysis directly from that. And actually, in that kind of dry blood spot, you can detect up to 800 different metabolites from one single spot. You have to present your sample to the instrumentation, and you do that with the different fashions. One way is to give it to the instrument by a liquid phase, so you have a thin capillary here, very thin glass tube. It sprays the molecules, body fluid for instance, into the instrumentation. Or you put small droplets on a surface like this, so you separate your molecules and then let them dry. Or you can actually take a whole tissue, a biopsy, or in this case it's a brain tissue, and you can make a thin slice of the tissue and then place that into the instrument and do chemical microscopy, you could say, on the tissue. So you can actually locate where is molecules present and how much of them are there. Mass spectrometers are fantastic instrumentations, and I will just show you a bit what's going on. You have instrumentation with strong magnetic fields, and you will have your ions, your analytes, circulating in this strong magnetic field, and then you will get the possibility to calculate the molecular weight of your molecules. And another format is this one, where you spin around the circular electromagnet, and you get your mass from the calculation. For those who are not following ion physics, this is in Swedish, so it tells you exactly how mass spectrometry works, actually. So how it is constructed, I will tell you at another time. It has to do with molecules and magnetism, and that's absolutely true, actually. So the problem we are facing is that some of the molecules that we are interested in are at extremely low levels, so we need to really push the sensitivity down. If we are looking at clinical tests, most clinical tests are possible to measure molecules in here, in this upper range of concentration. But as you see here in this picture, you see that we want to push this sensitivity down so we can actually measure at very low quantities. And one goal, maybe, in the future is to be able to measure one single copy of a molecule in circulation, which will be very hard, I agree. For those who are not into chemistry, I just want to show you an example how to calculate what we actually will be able to catch in a biological fluid. So this, let's assume we have a cup of coffee here and we add 1 lump of sugar in that. That will actually generate the concentration in the coffee of sugar. That would be 0.03 molar, and that is mole per liter. Do you remember what a mole is? No. 6 times 10 to the 23 molecules per liter is Avogadro's number, if you remember that from school. So a very, very large number of molecules, but we have it in a concentrated form now. So let's assume we take this coffee cup and we bring it to the swimming pool for some reason, and we drop it in the swimming pool and we let it dissolve in that swimming pool. Then we suddenly get into a low concentration of nanomolar. Then we take the coffee cup, bring a cup of water from this one, drop it in another swimming pool, and yet another swimming pool, and then take a sample from that. And then we are down to femtomolar concentration, and that's what we are going to measure. You understand, life is not easy in analytical chemistry.
Too many swimming pools.
Yeah. And then everybody who's been to a swimming pool, you know that there is not only water in the swimming pool.
Absolutely.
It's like, yeah, I won't say what it is. It's not very clean. So that makes things complicated. The liquid of interest that we are looking at is cerebrospinal fluid, and cerebrospinal fluid is generated and produced centrally in the nervous system in a gland called choroidus plexus. And we all produce quite a lot of this liquid, 500 to 600 milliliters per day. And during my presentation here, you will produce enough for me to do analysis on all of you, actually, so that would be perfect. We have a total volume of 150 milliliters. And how is now this possible? You have a production of 500 to 600 milliliters, but total volume is only 150, because we are resorbing the liquid. So it's produced centrally, and then it resorbs along the central nervous system, along the spinal cord. So we can actually, when we do a measurement of the cerebrospinal fluid, we will get a possibility to look into the central nervous system and the chemistry in the central nervous system. It is an invasive technique when you draw the lumbar puncture, and I think some of you have been through this. I made it on myself, actually, just to be able to tell that it's not so bad. I don't think it was so bad, but it wasn't pleasant, I have to say. But you get the local anesthesia and you draw the liquid. Sample handling is super important, so we are very careful with the samples. It's precious for us, and it's also precious for the patients who donate the material. So we have a strict protocol for this, so we can collect and make a standardized collection device for studies around the world, and also to sample, collect biobanks of the samples, and then also be able to transport them around the world. We have done some studies on over the years, and one study was to actually map the central nervous system healthy individuals, in this case, in cerebrospinal fluid. So we collected several hundred patients or controls, healthy controls in Sweden, and also collaborated with groups in US. So we mapped the normal human cerebrospinal fluid, and that was a very rich liquid, so 3,000 to 4,000 proteins present. We follow this up. Well, the paper is not so exciting, maybe. It's a long paper, 776 pages long. But you can use it as a blueprint of what is normal and healthy, and then we can compare this with patients with different disorders. We followed up a couple of years later on a study where we compared cerebrospinal fluid with, from patients with post-treatment Lyme disease and with chronic fatigue syndrome, ME. And this has been a paper that's been downloaded a lot, as you see. I think maybe some of you in the audience have downloaded it also 29,000 times. That's a record, I think, for me at least. What did we find then? Well, we found in the chemical patterns that there is actually a difference between healthy individuals. This is a so-called Venn diagram. So if you have the overlap, it's the same quantity and the same molecules present. So here in the center, we overlap all the different groups we are looking at. But the outer ends here, you see this is unique for the controls, healthy individuals. Here we have the chronic fatigue ME patients, and here we have the post-treatment Lyme disease patients. So we find unique markers in the liquid from these different groups, so we can actually tell a bit about what's going on. To cut that story short, what we found was a low-grade inflammatory reaction in the patients with chronic fatigue and also with the post-Lyme disease. We also found dysfunction in the axonal guidance, so repair system in the central nervous system, and that was actually not working as well in the CFS patients. And then finally, interesting signaling pathway was activated that we actually find the same pathway in neurodegenerative diseases like Parkinson and Alzheimer. So recently, we have been trying to follow this up in Sweden, in the Swedish cohort, and the things I'm going to present now is not published, so please don't spread it so much. We have, well, there's different figures coming out, 40,000 patients potentially in Sweden, 20,000 patients, and yesterday I spoke with a Swedish representative, and he said 8,000, so at least a lot of patients in Sweden. We managed to collect in a cohort of the west coast of Sweden, 20 patients, 13 females and 7 males. Median age was 45 years of age, and disease duration, you see 9 years, and then the mental fatigue scale was 24 in the median. All the lab tests that were done on these patients were normal, more or less normal. There were nothing really dramatic. So that shows you a bit of the problem we have, that the lab tests, the clinical routine tests, are not sensitive enough, in some cases at least. We compared these patients with our control groups, over 200 healthy individuals, 50-50 males and females, and median age is 43 here, and their tests were normal. So what did we find? Well, in the patient cohort, and it's a small cohort, it's 20 patients, so it's hard to draw significant conclusions, but significant changes that we could find deal with neurological diseases, metabolic diseases, and immunological diseases. And this confirms our study that we did in the US material also. You can look at these data in many different ways, and one way is to see in this kind of volcano plot here. It shows you all the proteins that are normal. They are in a big bunch down here. You don't see how many there are. But those that are different and significantly different comes above the red line here. These are proteins that are significantly downregulated and upregulated. Another way of looking at it is that you use the patients as their own label, so to say, and then do principal component analysis like this. So here we see the controls. All of them are compressed into a green spot here. And then we see all the patients, the 20 patients, that are significantly different from the controls. We also got later on some solitary patients that we added on to the analysis. And interesting enough, they actually ended up in the same region of this analysis. So there is a pattern that we can sort of reproduce within the different patients. The data gives you a lot of information. You give protein names, you give quantities and modifications, and so on. But you can also use the information to try to understand what's going on in these protein changes. So you can do what we call upstream analysis, and that means what are known in literature and in science today that would explain the changes we see. So what we found in literature and using software for this upstream analysis, we found that, for instance, regulation of IL-6R, the receptor for IL-6, and IGF-1, together with disturbances on BDNF, HRAS, and CGAN, and PRL, all can be explained by the changes we found on significant levels on proteins here. So this kind of network can tell you a bit on what could be the reason for the changes we see in the proteins. And the same way, if we look at downstream, so what could the result be? If you have a protein change in the system, what would that give in the end? We can see that the results show you an increase in cell activation and carbohydrate regulation, while decrease of concentration of certain lipids and the chemotaxis of myeloid cells is changed. So we can see that there are things that actually gives us an idea of what's going on based on the proteome. So conclusion is that we have successfully found and repeated some of the findings we have from the American study in this small cohort, Swedish patients. We found some new potential markers that can be used, and we also got some new hypotheses on things that are changed in the ME CFS patients. For instance, glycoprotein disorder, lipid dysregulation, that can relate to energy deficiency and fatigue. So I want to acknowledge my collaborators, and in this case, it's the Gottfried's Clinic in Mölndal in Sweden, who has been helping us to collect the samples. Katarina has made the analysis, and then Gia, who is my bioinformatician. And bioinformaticians are needed a lot in this kind of research because we generate thousands and thousands of data points, and we need to understand how they are all working together. And also a warm thank to all the patients and the controls that have, on altruistic basis, donated sample to this kind of research. And I thank you for your attention.
Thank you, John. We have time for a couple of questions, okay? Would anyone like to open up and ask a question? Yes, please.
One of the big difficulties that the medical profession who believe in the organic hypothesis of the causation of this condition is they're always opposed by the psychological paradigm. And my psychiatric colleagues will always say that these changes you're describing are downstream, in other words, the effects of primary psychological disorder. What argument do you use to oppose that?
Yes, that's a bit of that's a trouble actually, because you can of course assume that certain psychological effects would even, change protein levels and change patterns like this. So it's sort of a problem to actually address when you want to get back on that. But if you look at the, that's why it's important, I think, to not only look at the patterns you see, that you want to tie it to biology again. So look at what are the effects really. Is it immunological effects, or is it cell repair systems that are affected and so on? The psychological role part of it can maybe not be excluded totally anyway, if you look at those phenomena. But it's a very complex regulatory mechanism that is affected. So what is good with the kind of unbiased analysis that we have is that you can exclude, so to say, the measurement that will not give any, or you see the clear-cut differences in the biochemical unbiased assay. So you can use that as an argument, I guess, for that something is really going on. And even if the routine clinical chemistry laboratory tests don't show anything, with more sensitivity technology, you can actually find differences.
Would that convince them?
I'm not totally sure, but
I know some who would never be convinced.
Exactly.
I think you have to be rather open-minded. I think there can be a lot of different effects going on, of course.
My own view is that what you're describing is science, and what the psychiatric opponents to the proposition that this is organic disease is that they are reaching for a doctrine which no longer applies, in my view.
Okay. Anyone else? Quickly? Yes, ma'am.
The recent American review, one of their criticisms of a lot of research was that the control groups were not wide enough. In your work, are you, for instance, considering putting in lazy or at least not active individuals and people with depression as controls to compare with your samples from the ME community?
Our controls are, I think, we are very lucky to have the chance to get these controls that are totally healthy individuals, but with a large age span. So we have, 16 to 65 years of age, for instance, it's the cohort that we use for our controls. And they are truly healthy individuals that comes on altruistic basis to give a sample. So many other studies that are done in cerebrospinal fluid is done, then they use other controls like headache patients, migraine patients, patients with unclear diagnosis and so on, and they don't find anything on the routine clinical tests, and they assume that they are normal and healthy. And we can compare our healthy controls with our true healthy controls with, for instance, headache patients, and we see big difference in proteins, especially proteins that relate to small bleedings and circulatory dysregulation and so on. Okay. I'm not sure if I answered your question because I didn't hear totally the last part.
Okay, well, thank you very, very much, Jonas, and safe journey back.
Thank you.